Okay, so I've been getting a lot of feedback on the various VDJ study materials you've received from anywhere from Robert May to Leah Smith. So I thought I would create a video that kind of goes over the whole thing, explains it a little more so that you guys can kind of see where the details are important and where really just a global understanding of how antigen receptors are formed will carry you through the material. And we're actually going to do this using the mechanisms of diversity, because really that's the whole point of VDJ, is creating diverse antigen receptors. So we have 5 genetic mechanisms of diversity. You will be covering somatic hypermutation beginning in Leah Smith, and maybe a little bit in Katie Diaz. But in the initial cases of Robert May and Jenna Wilson, you only knew about the 4. So that's inheritance, recombination, recombinational inaccuracy, and random assortment. So the first thing that we have to think about is anytime that we are talking about VDJ, we're talking about T-cell receptors and B-cell receptors. So let's start first by just looking at their structure. So when we talk about a B-cell receptor is basically just an antibody that is stuck on its surface. Okay? And then an antibody has a heavy chain, as I've drawn here, and then it also has a light chain. Okay? Then when we think about the different portions of the antibody, you've got the areas that bind antigen, which are like right here and right here. But then you also have the portions that allow it to be attached to the B cell, so determining whether or not it's going to be membrane bound or if it's going to be secreted. And then you also have the portion of the antibody that's going to determine what class of antibody it actually is. So this tells you whether or not it's like an IgM or an IgG. So that's just to kind of orient you to the structure, really, of the actual antibody molecule. Okay, so when we talk about T-cell receptors, it's actually not that different. It's almost like you took this little piece of the B-cell receptor, just this antigen binding piece, and moved it over here to be a T-cell. Now, obviously, they're very different in structure, but the concept is the same. So in the B-cell, we have a heavy chain and we have a light chain, right? And that's what I showed here. With T-cells, we actually have an alpha chain and a beta chain. So that's kind of what I'm drawing over here. So your alpha chain would be like here, and your beta chain would be like here, okay? And then they stick through the T-cell, right? Because they're always surface bound. So we don't have quite the same level of complexity in the structure on the T-cell receptor. Now, what are we actually talking about with VDJ? With VDJ, we're only affecting the part of the protein that is going to come in contact with the antigen. So for T-cells, we're talking about this area right in here that's actually going to bind our antigen of interest. Okay, so if this is where our antigen is going to bind, we only care about the part that's actually touching our TCR. And the same can be said on the B cells. So we're only talking about this part right here where the antigen is actually going to bind. We're not talking about the rest of the molecule. The rest of the molecule is already encoded for, accounted for, and we know what it's going to look like. And this is especially true for T cells, because remember, with T cells, these areas right here, they need to be able to bind to and recognize self-MHC. That's kind of their whole deal. If they can't, in these invariant regions, recognize self-MHC, then they can't see the antigen. It doesn't matter if the MHC is presenting it. So we're really only affecting these areas where the antigen is physically touching the T-cell receptor or the B-cell receptor. That's the whole point of VDJ rearrangement. And the reason for this is that we need to make an unconscionable amount of antigen receptors. If we had to encode for every single antigen receptor, we would need, over the course of our lifetime in our DNA, it would overwhelm the human genome. It would be way more than the human genome because there are just so many unique antigens in the world. We would cease to be humans and just be antigen recognition proteins. So instead, we've made building blocks. We have building blocks that we use to create these unique locations within our TCR or BCR. So how does this work? I want you to think of it for the moment very simplistically, okay? So this is our chromosome, and it has a whole bunch of genes on it. And the first thing we're going to note about it is that it literally has multiple copies of the same gene type, but they're each a little different. So it has multiple V regions or V exons. It has multiple D exons, and it has multiple J exons. Okay? So when we think of it that way, when we think about how many we have, that means this is V1, 2, 3, 4, 5, because these are all of our V genes. And this is D1, 2, and 3. These are our D genes. And this is J1, 2, 3. These are our J genes. I don't know exactly how many there are. I think I listed them in your self-study, but I don't really care. The idea is that we inherited a whole bunch of them from mom and dad. Thanks, mom and dad. So what happens is we can use these to create our unique receptors, and we do that by recombining them in a seemingly random order. Okay? So we're going to start with looking at both TCR and BCR, because really the process is the same, just the names change, okay? So for a TCR, we're going to start with the beta chain. For a BCR, we're going to start with the heavy chain. What we're going to do is we're going to choose, say, D2 and J1 and put them together with, say, V1, okay? That is a rearrangement of either a beta chain or a heavy chain on this cell, okay? We took one V region, one D region, and one J region, and we made a chain. Great, that'll work. And then let's say another cell made V2, D3, J2. This one is going to form part of the specific structure for antigen A. And just by virtue of it being a different exon, this one is going to form part of the structure that will be specific for antigen B. Because we used completely different V, D, and J regions, we have different specificities because they're going to be slightly different, okay? So let's say, though, that that second cell also used V1, D2, J1. Now, you would think that looking at this, it would also be specific for antigen A, but you would be wrong, and here's why. It's because of recombinational inaccuracy. This is an incredibly inaccurate process. These cells are replicating very quickly. It's error prone, and we like it that way. Why? Because errors bring diversity. And the other thing we have, particularly in the beta and heavy chains, is the enzyme TDT. TDT adds your N nucleotides. So it's going to add N nucleotides anywhere you have a joining. So that means this V1, D2, J1 is actually V1 with, a couple nucleotides here, and D2, couple nucleotides, J1. This one is going to be a totally different set. And just by virtue of having these extra nucleotides, now we're back to this one being specific for antigen A and this one being part of the complex that will be specific for antigen B. So once again, we've increased our diversity. So all we've done at this point is made a beta chain or a heavy chain, and already we've effectively worked with 3 of the big methods for creating diversity. We inherited a whole bunch of different V, D, and J segments. We reorganized them randomly, kind of like boxcars on a train. And then we actually did kind of a bad job of it because we replicated it weirdly and added in random nucleotides using TDT anywhere we wanted, but that's great because it increased the diversity even more. And it made that even though we were using the same V, D, and J regions, we actually have a different specificity for a different antigen. Okay. So just doing that was a lot of work, right? So we successfully made on just one chromosome a VDJ, and that is either going to be a heavy chain if we're talking about a Bcell or a beta chain if we're talking about a Tcell. Because it's so much work, we don't want to waste that. We don't want to have to do it again. We are 50% of the way through making a functional antigen receptor. So we're going to check it. We're going to do this in two different ways. If it's a Bcell and it's a heavy chain, you're now going to enter that large pre-B stage, right? So that means this chain is going to be expressed within a vesicle in the cytoplasm of the cell, and it's going to be expressed the V pre-B, right? These are basically your training wheels. It's just testing if the heavy chain it made can successfully bind with a light chain if it was to make it. That's all it is. It's like we haven't wasted our energy making the light chain yet because light chains are really energy expensive to make. Before we go ahead and do that, let's make sure the heavy chain we made could actually bind with a light chain. If we're talking about a T-cell, it's a similar process. In T-cells, they're going to take their beta chain and they're going to put it up on the surface and they're going to pair it with what's known as pre-T alpha. Pre-T alpha is basically the equivalent of V pre-beta. And all it is once again, it's some training wheels. It's a surrogate light chain that's just going to prove that the beta chain it made will be able to successfully bind with an alpha chain if we went ahead making it. Here's the great thing. How do we actually know that these chains that we made are good chains? Well, it's going to actually signal through it. We're going to get a signal through these chains. And if it signals, it's going to tell the cells to proliferate and proliferate wildly. So suddenly, we're going to have a whole bunch of cells that all have the same either heavy chain or beta chain on their surface. They're ready to go. Well, actually, in this case, it's actually going to be intracellular if we're talking about a B cell. So it'll actually be in here. But you're going to have a whole bunch of cells that have this heavy chain inside them, no light chain yet, or a whole bunch of cells that have this beta chain on the surface. And the reason is we already made this chain, and it took a lot of energy. And it's a good chain because it caused proliferation. So let's use it. Let's not just use it for one cell. Let's use it for a lot of cells because it's a good chain. Let's become a good chain building factory. But you're like, wait a minute, if I'm using that same chain, isn't that going to affect my diversity? No, because we haven't made our light chain or our alpha chain yet. So what happens is after we make our light chain or alpha chain, all of these cells are going to make a different light chain or alpha chain. And that's going to change their diversity as well. This is called random assortment of chains. And all it means is that we took the beta chain or the heavy chain that we made over here and we're pairing it with lots of different light chains or alpha chains. Okay? That's the whole thing. So we want to do that. We haven't done it yet. So we went over here and we proliferated the cell, and all it has is our heavy chain or beta chain. So we've got VDJ, and we made that. That's good. Now we have to go ahead and make a light chain or an alpha chain. If we're talking about T cells, this is pretty easy. It's going to go and it's going to choose a V region and it's going to choose a J region. And it's going to go ahead and choose those. And what you're going to have is you're going to have your protein here. This is my crudely drawn T-cell receptor. This is my beta chain. Let's say it's V1, D1, J1. and this one is V4J3, okay, because this is my beta chain and this is my alpha chain. If the alpha chain is functional, it will bind with the beta chain, and then since it's a T-cell, it's going to go through positive and negative selection. So positive selection, remember, is it needs to see MHC. Negative selection is it can't see self-antigen. If it recognizes self-antigen, it dies, okay? If it can't see MHC, it'll die, all right? And that's it. That's the whole show with T cells. They're pretty simple. Everybody look at that because I'm about to erase it. Good? Okay. Now, if we're talking about B cells, it gets a lot more complicated because B cells are kind of the problem children of lymphocyte development. So if we're talking about B cells, they go ahead and they do the same thing. So you have your heavy chain, which was B1, D1, J1, B1, D1, J1, okay? And now we need to make our light chain. So let's say for our light chain, it chooses V3, J2, okay? And then it goes to test it to see, okay, is it reactive to self-antigen? Let's say the answer is yes. If the answer is yes, it goes, wait a minute, just kidding, I can fix this. And it goes and it chooses a different V region. So now it's going to go like V5, J2 and V5, J2, okay? It'll keep checking for self-antigen, and it will keep rearranging this B cell receptor until it either makes a light chain that when paired with the heavy chain it had already made does not recognize self-antigen, or until it runs out of genetic material. If it runs out of genetic material, the cell dies, and there's nothing that can be done about it, okay? All right, so let's say it does this and it makes a good pairing. Then you'll have a B cell that basically is a nice heavy chain on the surface, and also has a light chain that is not self-reactive. This is always going to have the IgM backbone, and this is what we would call an immature B cell, okay? It's going to check this. If it checks it and all works out well, it's going to allow for co-expression of an antigen-specific identical IgD on the surface. So how does that happen? So I'm going to redraw something down here that I have up there. So if we look at our gene region, you know about the V regions, and you know about the D regions, and you know about the J regions. But there's more. There are class regions, 5 of them to be exact. So those would be your constant regions. So it's C mu, C alpha, C gamma, C epsilon, and C delta. And those basically translate to IgM, A, G, E, D. Okay? Because those are the different types of antibodies we have. But wait, there's even more. The B cell has to determine if what it has is going to be secreted or membrane bound, right? Because the immunoglobulin is the B cell receptor. So you have domains here for secreted versus membrane bound. So a true B cell receptor looks something more like this. This is our heavy chain, and this is our light chain. Then we have some red representing our V regions, some green representing our, whoops, never mind, not there. I'm trying to make it colorful so you can follow along. All right, some green representing our D regions only on the heavy chain, and some blue representing our J regions on both the light and the heavy chain. So that's what we've done so far. The rest of this is all backbone. So remember, we're making this B-cell receptor for the very first time. So it's automatically going to have C-mu, okay? So this backbone becomes C-mu, all right? So that's what it actually looks like. Then you have to choose membrane bound or secreted. Well, this is still a B cell. In fact, it's a very new B cell. So it's going to be membrane bound because it's going to be attached to the B cell membrane. Okay, so you've got membrane bound and you've got C-mu. So you've got IgM bound to the cell surface. Okay, now once it's done this, it gets signaled here to go into its nucleus and allow for co-expression of IgD. So that's going to be antigenically identical. It's just also going to be on the surface. This process that allows for co-expression of IgM and IgD is a result of alternative splicing. And alternative splicing occurs at the RNA level. And that's important because what we're doing here is we're allowing for the expression of C-mu or C-delta, but we haven't ruled out the possibility that one day this B cell will express alpha, gamma, or epsilon. We also haven't ruled out whether or not it will be membrane bound or secreted. We're not making any hard and fast decisions here. All we've done at this point is determine that we are ready to leave the bone marrow and go and circulate around the body and hopefully find our antigen of interest in the lymph node. Okay? All right. So let's do that. Let's leave the bone marrow. Out we go. So now this young B cell is off in the great big city of the lymph node. And let's say it found its antigen of interest. Okay, so it's got its B cell receptors on the surface, right? Because it was allowed to leave. Because IgD is just kind of its diploma. D is for diploma. It lets them leave home. It lets them leave high school so that they can go on to college. Okay. All right. So now we're in the lymph node and we see our antigen of interest. On top of that, well, let's start small. We see our antigen of interest. Now, something's going to happen. This cell is going to do a couple of things. One, it's going to differentiate into a plasma cell. And the first type of plasma cell it's going to be is an IgM secreting plasma cell. So it's going to go from just having IgM on the surface to now actually making IgM that can then travel around the body and bind up whichever pathogen it encounters. Okay? IgM is a pentamer, in case you guys hadn't figured that out yet, but I think you did. It's in your Leah Smith notes. Okay, so how does it do that? Well, it does that again by alternative splicing. So once again, we're going to go up here, and instead of choosing membrane bound, we're going to choose secreted for this last portion of the receptor. And once that's turned on, some IgM will remain on the B cell surface so that the B cell can continue to be activated by its pathogen. But some of the IgM that's produced will be secreted because it will have the secreted kind of signature, okay? The other thing that could happen is that this B cell could see its antigen of interest in the context of a T cell. Okay, and let's say it's going to present some of this antigen to this T cell, okay? The T cell then will secrete some cytokines that give some instructions to the B cell. On top of that, you're going to have CD40 ligand interaction. And the CD40 ligand interaction is going to activate an enzyme in the B cell known as AID. AID is going to make some real changes around here. What it's going to do is that it's going to tell the B cell to not only differentiate into a plasma cell, But this time, because it's had T-cell help, it's going to say from now on, instead of making IgM, we're going to start making IgG. And all we make is IgG. Sorry, the one on the surface remains the same. It's just the B-cell receptor. But this run out here, these are all going to be IgG. And it's never going to make IgM or any other class of antibody again. This process is known as isotype switching. Isotype switching is a permanent change. And anytime you have a permanent change, we're talking about a DNA level cut. So what happens is AID comes over here and it cuts out all of the DNA that it's never going to need again and only keeps the one that it's signaling via the cytokines to express. So that's kind of the role of AID. And why I care about it is that there are patients who lack AID. And when a patient lacks AID, they lose the ability to class switch. And you might not think that's a huge deal, but think about it. It's kind of similar to hyper IgM. So their expression of AID will affect how many different classes of antibodies they get. And they're more susceptible to different types of infections because as you're learning in the Leah Smith self-study, IgG has specific functions. IgM has specific functions. IgA has specific functions at the mucosa that protect those sites from, different types of microbes that we see there. So we have to keep thinking of these antibodies and the functions they have when we're not able to class switch. Okay, so let's just go back up here and look over some big terms, and then I'll leave you guys alone because I've been yammering for a little while. So you have 5 genetic mechanisms of diversity. I didn't cover somatic hypermutation here, but I think my video for this one is pretty good, so check it out. It's in the Leah Smith study guide. We did talk about 4 of them, though. You inherit multiple VD and J regions. You recombine those gene segments to make unique receptors. And if that wasn't enough to create diversity, we also promote diversity here. We promote mistakes. We promote changes and different expression of proteins. That's what makes the immune response strong. And TDT is a big player for that. And it only makes changes in the beta chain or the heavy chain, but it's still enough to change the antigen specificity of that lymphocyte at some point. And once you make a chain and you know it's a good chain, pair it with a whole bunch of new ones. So that's that random assortment of chains. You learned about alternative splicing, which is an RNA change. You're keeping your options open, so you can still make IgG later. You can still make a secreted or a membrane-bound antibody later. But we also learned about isotype switching, which is a DNA change and is a lot more permanent. All right, let me know if you have any further questions on this. Good luck.